endothelial to mesenchymal transition is a cellular process in which endothelial cells change their cobblestone morphology into a fibroblast-like phenotype. And the cytokine TGF-β, transforming growth factor beta, is a main driver of this transition.
Analysis of the changes in cell morphology and the expression levels of endothelial and mesenchymal markers allows us to assess the occurrence of endothelial to mesenchymal transition.
Emerging data link the endothelial to mesenchymal transition process to various diseases, including cancer, cardiovascular disease, and fibrosis. And the data suggest that the intervention may be of therapeutic benefit. By stimulating the endothelial to mesenchymal transition process, which occurs during embryogenesis and organ development, we may possibly be able to regenerate mesenchymal-derived tissues, such as cartilage, bone, and muscle.
To detect the intercellular protein by immunofluorescence staining, cell permeabilization using 0.1% Triton X100 is necessary to allow the antibodies to pass through the cell membranes. With this visual demonstration, researchers will get a good grasp of how to investigate the role of cytokinase, such as TGF-β, in endothelial to mesenchymal transition.
First, seed 1 × 10⁵ murine pancreatic islet endothelial MS1 cells in DMEM supplemented with 10% FBS and 100 units per milliliter of penicillin and streptomycin onto a 0.1% gelatin-coated culture plate.